All NEET aspirants, you are listening to the podcast Botany for NEET by S. Ramakrishna. In second episode, the molecular basis of inheritance, the structure of polynucleotide. Polynucleotide, that is many nucleotide linked to form a polynucleotide, which is nothing but polymer. So building blocks of nucleic acids are nucleotide. In DNA, deoxynucleotide. In RNA, ribonucleotide. Next. In nucleotide, there are three components: nitrogen base, sugar, phosphate. In nitrogen base, adenine, guanine, thymine, cytosine, uracil. So, five types of nitrogen bases. Next, adenine, guanine, which are purines. Thymine, cytosine, uracil, which are pyrimidine. Both the purines and pyrimidines, which are having carbon nitrogen rings, what do you call heterocyclic compound. And purines are dicyclic, having two rings. And pyrimidines are monocyclic, having one ring. And dicyclic compound adenine and guanine, which are 9-membered ring. Thymine, cytosine, uracil, which are monocyclic, which is having 6-membered ring. In dicyclic compound adenine guanine, 1, 3, 7, 9 are nitrogen position. In case of pyrimidine, like thymine, cytosine, uracil, 6-membered ring, 1, 3 are nitrogen positions. Next, adenine, guanine, which are common for both DNA and RNA. So purines are common. In pyrimidine, cytosine is common for DNA and RNA. So what are common for DNA and RNA? Adenine, guanine, cytosine. Which is not common? Thymine, uracil. Thymine present in DNA, absent in RNA. Uracil present in RNA, absent in DNA. Next, adenine. 6-aminopurine is adenine. Thymine. 2,4-dioxy-5-methyl pyrimidine is thymine. Guanine. 2-amino-6-oxypurine, it is guanine. Cytosine. 2-oxy-4-amino-pyrimidine is cytosine. Uracil. 2,4-dioxypyrimidine, it is uracil. So uracil differs with thymine by lacking methyl group. Next, and you have to check here which is having amino group, which is having oxy group, which is having both the amino as well as oxy group. That constitute one question. Next, bond, sugar and phosphate. In case of sugar, it is a pentose sugar, deoxyribose sugar, C5H10O4. There is a deficit of one oxygen. That's why it is 2-deoxyribose. Ribose sugar, C5H10O5. So both are pentose sugar. Phosphate. Phosphoric acid biologically called phosphate. As oxygens carry negative charge, that's why DNA it is negatively charged. Next, bonding. Nitrogen base attached to OH of the first carbon through N-glycosidic linkage. 9-1 linkage in case of purine, that is the 9th atom of nitrogen, first carbon sugar of sugar. 1-1 linkages in case of pyrimidine. So 9-1 purine, 1-1 pyrimidine, that constitute glycosidic bond. With this glycosidic bond, it forms nucleoside, deoxyadenosine, deoxythymidine, deoxycytidine, deoxyguanosine. In case of RNA, adenosine, guanosine, uridine, cytidine. Next, and phosphate attached to OH of the fifth carbon by means of phosphoester bond that forms nucleotide. It is a mononucleotide. In a mononucleotide, one nitrogen base, one sugar, one phosphate, one glycosidic bond, one phosphoester bond. Next, in case of nucleotide, in case of DNA, 4 types of nucleotide. In RNA, 4 types of nucleotide. So there are How many types of nitrogen bases in nucleic acid? 5. Adenine, guanine, thymine, cytosine, uracil. How many types of nucleosides in nucleic acid? 8. DNA 4, RNA 4, because deoxyribose sugar, ribose sugar. How many nucleotides in nucleic acid? 8. DNA 4, RNA it is 4. Next. Adjacent nucleotides held by phosphodiester bond. 3,5 phosphodiester bond. So it is a dinucleotide after formation of phosphodiester bond. In a dinucleotide, how many nitrogen bases? 2. How many sugars? 2. How many phosphate? 2. How many glycoside bonds? 2. How many phosphodiester bond? It is 1. Opposite nucleotide held by hydrogen bond. So on one strand, the phosphate moiety at the 5 end, that is called 5 end of polynucleotide. On other end, 3OH, the on sugar, the free 3OH, so it is called 3 end of polynucleotide. Overall it is 5-3 direction, that is showing what it is polarity. Next. In other strand, it is 5-3 means it is 3-5, it is anti-parallel, opposite direction. In DNA, sugar phosphate sugar that form external backbone and nitrogen bases extended from external backbone more or less perpendicular towards inside. Next. In every ribonucleotide residue, there is addition of one oxygen at second carbon that makes the difference with the deoxyribose sugar. Next. Nucleic acid discovered by Frederick Miescher called as nuclein, having acidic properties. And elucidating the structure of DNA was elusive for number of years, for a long time. Why it was elusive? Because there is a technical limitation. What is the technical limitation? Extraction of a long polymer is difficult. That too, it is an intact polymer, intact with carbohydrate, protein, as well as lipids and RNA. So, extraction of a long polymer is a technical limitation. That's why it took long time to decipher the structure of DNA. Next. Watson and Crick able to propose a fabulous double helical model of the DNA. That too after observation of Wilkin and Franklin, their data. With the help of the data given by Wilkin and Franklin based on X-ray diffraction, Watson and Crick able to propose double helical model of DNA. Next, the hallmark of excellence of Watson and Crick is base pairing. That too, after observation of Khargov, purine and pyrimidine exist in 1:1 ratio according to Khargov. A plus G is equal to T plus C, or A plus G by T plus C is equal to 1. So, A plus G is equal to T plus 1 is universal in all DNAs having double-stranded DNA, but not a single-stranded. Suppose in 120 nitrogen bases, 10 adenine means 10 it is thymine. In remaining 150 guanines, 50 cytosines. So 10 adenines, 50 guanines, 60 is equal to 10 thymine and 50 cytosines. So A plus G 60, T plus C 60, 1:1. It is universal, common in all double-stranded DNA. AT-GC ratios varies and it is species specific. If you take bacteria, GC ratio more than AT ratio. That's why more number of hydrogen bond it is stable. In case of eukaryote, AT ratios more than it is GC ratio. That's why it is less stable compared to prokaryote. Next. And unique property of the DNA is complementary of base pairing. If you know the sequence of nitrogen bases in one strand, you can predict the sequence in case of other strand. Suppose in 5,3 ATC, in other strand it is 3,5 A adenine pairs with thymine, thymine pairs with adenine, and cytosine pairs with guanine. Purine pair with pyrimidine, pyrimidine pair with what it is? The purine. That is one point. Next, genetic implications with the structure of DNA became so clear after proposition of replication of DNA. Genetic implication, the doubts with regard to the structure and genetic material, it became so clear after proposition of replication of DNA. How it is possible? DNA is a double-stranded molecule. After replication, two daughter DNAs are formed. The two daughter DNAs are exactly similar to parent DNA because in DNA, two strands act as template. On each template, a new strand is formed. So, how many total daughter DNA form? Two daughter DNA. So, genetic implication with the structure of DNA as a genetic material became so clear after proposition of replication of DNA. The doubts cleared after replication. Two daughter DNA exactly similar to parent DNA. Why? Two strands act as template. So molecular weight of each daughter DNA similar to that of parent DNA. Molecular weight of both strands are not same. Molecular weight of two each daughter DNA similar to that of the parent DNA.